All right, we are back. Oh. All right, let's wait a sec while I can see streaming on Twitch is working, but right now we're waiting for... Ah, there we go. Okay. We're streaming in both locations. All right, nice. Okay. So yeah, we're changing things up a little bit. So I figured since I'm very interested in the sciences, and I like the sciences quite a lot. I figured why not start off with actually reviewing some papers. So I figured, so that's exactly what I'm going to do. It's going to be fun. At least I'm going to find it fun. All right, so what we're going to do today is we're going to look at this particular paper right here. Inhibition of fatty acid oxidation enables heart regeneration in adult mice. This was published in Nature in 2023, so it's actually very recent, extremely recent actually. It's not even a year old. And in science, recent is usually classified as like 5 years, 10 years. So yeah, this has practically just been discovered. And it's already quite interesting. Just the title alone has me scratching my head because heart regeneration, right here, heart regeneration in mice. You might not know this, but most mammals, and I mean a lot of mammals, they have trouble with heart regeneration. It's like in humans, if you end up having an electric shock to your heart is can be effectively messed up for the rest of your life. The heart is very, very keen on not being disturbed. And here they actually have talking about in the title that you can promote regeneration, something I previously thought was only possible in certain organisms like axolotls and zebrafish and salamanders. So it's quite interesting. And they claim that it's involving the inhibition of fatty acid oxidation. So we should probably go through here and check out the data, see what it shows off, and figure it out. For every paper, first thing you gotta do is first thing to do on the paper is we always check the abstracts. So let's see here. So postnatal maturation of cardiomyocytes is characterized by a metabolic switch from glycolysis to fatty acid oxidation, chromatin reconfiguration, and exit from the cell cycle, instating a barrier for adult heart regeneration. And they even have references here. Let's see here. Let's see here. Cardial maturation. Yeah, I am not a cardio. I don't study the heart. That's not my forte. But whenever you're studying research, you tend to find information from all sources. You don't pick and choose. You just look at everything. So let's see here. So it's interesting to see is that... All right then. So we have a switch from glycolysis to fatty acid oxidation, which will result in the exiting of the cell cycle. And this is a major barrier for heart regeneration. Here they explore the metabolic reprogramming can overcome this barrier and enable heart regeneration. We abrogate fatty acid oxidation in cardiomyocytes by inactivation of CPT1B. Ooh, a gene. Ooh, I wonder what. Ooh, whoops. Yeah, there we go. Okay, I wonder what this does. CPT1B. We found that disablement of fatty acid oxidation in cardiomyocytes improve resistance to hypoxia and stimulates cardiomyocyte proliferation. All right. So I guess I should start trying to explain a couple things here. So what does this mean? So disablement. So they deactivated the gene of the fatty acid oxidation. So they're prohibiting the cell cycle in cardiomyocytes. Cardiomyocytes are pretty much the cells within the heart. Cardio is heart, myocyte cells. And improves resistance to hypoxia. Hypoxia is pretty much the lack of oxygen and stimulates cardiomyocyte proliferation. So by disabling the fatty acid oxidation, it prevents, or it makes the cells more resistant to suffocating, and then results in proliferation and allowing heart regeneration after the ischemia reperfusion injury. So ischemia reperfusion pretty much means that you have blood start going back into the heart. Wow, this is a very complicated sentence here. If you don't understand the linguistics of a science paper, then yeah, this could be very you have a lot of words to look up. Anyway, so here we go. Metabolic study reveals profound changes in energy metabolism and accumulation of alpha ketoglutarate in the mutant cardiomyocyte. Ah, they used a mutant to downregulate. All right, interesting. Leading to activation of alpha ketoglutarate dependent lysozyme demethylase, KDM5. Activated KDM5 demethylates broad HK. Why do I have a feeling we might have to start looking some of these genes up? Myocyte maturation, lowering their transcription levels and shifting cardiomyocytes into the less mature state, thereby promoting proliferation. We conclude that the metabolic maturation shapes in epidemic landscapes of cardiomyocytes, creating a roadblock for further cell division. Reversal of this process allows for repair of damaged hearts. Ooh, interesting. So what they're proposing is that because of this increase, because of hypoxia and this increase in fatty acid oxidation, it creates a barrier or a boundary, a roadblock, which will prevent cell division from occurring, which will then result in decreases in the repair of heart damage. I'm a bit curious because if I recall, when it comes to heart damage, one of the biggest problems isn't necessarily the proliferation of cells, but it's the scar tissue formation. Oh, I wonder if it could be along the lines of the fact that we have a decrease in the amount of cells that are proliferating, it just results in scar formation taking over and fixing the problem. Because one of the big problems with heart damage is when you have damage to the heart, you run the risk of this very hard, and I mean, scar tissue is known as being very hard, very immalleable. It doesn't twist and bend and turn, a lot like you would normally find in anywhere that's not scarred. Anyone who's had a big cut on their arm and had a scar form there, you can attest to how difficult it is for a scar to do anything normal like normal skin would. So it's always been the case where heart damage results in scar formation, which would result in a heart that doesn't work very well. So I wonder if by increasing cell proliferation, it actually mitigates and decreases scar tissue formation. Well, let's see. I wonder if they talk about it in here. All right, so abstract gone through. The main is usually a little bit about papers. Usually when it comes to reading papers, the most important is the abstract. Then it's the figures, followed by the results, and then the discussion. And then once you feel like you're done, once you feel like, oh yeah, I think I've had enough, then you go back and read the intro. And you might be wondering, that's a bit weird. Why would you skip the main page? Reason being is that usually this here, the main, and the main, hypoxia, or actually I think this is one giant result, isn't it? Usually you tend to ignore like the main or the descriptor is because of the fact that it's all putting the paper into context. For instance, if you study the heart, you don't need to have a crash course in cardiomyocytes and heart damage and the problems that occurs when you have a damaged heart. You probably already know this, so most scientists tend to just skip that, and then they go back there to see if there's any references that they might have missed, to go read somewhere else. Usually you tend to already know a lot of information about this, so we're going to go ahead and skip this. I'm going to head straight into the figures. Oh, hey. Extended figures. Ah, nice. Okay. First things first, let's see here. Inhibition of CPT1 stimulates cell cycle activity and hypertrophic growth of neonatal cardiomyocytes. All right, let's check this paper. Let's check out this figure. All right. So let's see here. So the first figure on the... I just noticed it loaded up into a different window. Okay, maybe I can... 1 second while I fix this. Aha, there we go. Aha, there we go. Okay. Here's our figure right here. Let's go ahead and get on in nice and close. Ooh. All right then. So this happens to be expression levels. All right. Oh, can I hide? Oh, maybe not. I want to hide. Oh, there it is. Aha, there we go. Good, perfect. All right. So first things first, let's check out what these figures mean. So this is a heat map representing expression of genes involving glycolysis, cell cycle, fatty acid oxidation, and the Krebs cycle. All right then, all involved in energy process, the energy production. Publicly available RNA-seq data and CM isolated from mice. From these ages, adult mice 1 week after transaerobic construction were used. Construction. Okay, yeah, construction. A constriction. Adult mice 1 week after transaerobic constriction was used. So pretty much they took a bunch of RNA from hearts of mice and they looked at the different time points to check to see, they constricted the heart, so they caused ischemia or decreased blood flow in the heart. That's one way to damage. Okay. The claim is publicly available RNA-seq data, so that means you can easily get a hold of this data in the literature. So if you want to, you can look at it yourself. But this is pretty much all of the genes that have been published on and the expression levels. So let's take a peek here. So if we look at these genes, we can see that there's I wish they had a better magnification. Anyway, let's look here. Yeah, I wish a better magnification. But here are various different genes, and we can see that some of them are being risen, some are being dropped. You can see that here's the expression levels, negative 1.5. Interesting. It's a z-score. Interesting. All right. I can understand 1.5. When I was doing these studies, I always used 2. I always used plus and minus 2 because, I mean, 1.5 you can still find a lot of good genes and you can find a lot of good stuff. But at the same time, it also, the higher the z-score, the better, because the less likely this is just noise. As you get closer to 1, the more likely this is just basic noise. So, yeah, I think it's good that it's 1.5. It's nice, it's fun to see, but yeah, I usually, in papers, they usually go for the 2s. However, yeah, it's it's a complicated mess when it comes to these things. It's a complicated mess. Because the most important genes, sometimes they don't get regulated that high. You learn that quickly when you start studying cancer. When you're looking at cancer cells, you quickly find that, the STAT pathway is upregulated and, it will cause cancer. But then you find out, there are certain other genes that they're regulated just a little bit, and they play major roles in cancer. The oncogenes. So, yeah. Anyway. Hey, how's it going? Let's see here. We're having fun looking at a paper right now. So let's see here. There's an awful lot of genes here, an awful lot of gene expression. And here are all of the different age groups. And here we could see CPT1A, which is the gene of interest, is completely modulated differently. It's almost completely the opposite of a lot of the other genes within the cycle, the fatty acid oxidation cycle. That's very interesting. Well, of course they're going to target it if based off of this data right here. I'll target it too. Well, I mean, this is more along the lines of how to repair a heart. Uh, if you want development of a heart, a growth of a heart in that case, if you want to sell them, and actually there's quite an overlap when it comes to the two. It's quite an overlap. Either way, let's see. So yeah, there's that, and then here we have more expression levels between the different groups, and again, CPT1A is being upregulated. So yeah, pretty much all of figure 1, if we look here, is pretty much just showing the levels of CPT1. Yeah, and then they have lots of different controls. So okay, yeah. I believe him. The first figure, the showing that CPT1 is involved in cell cycle and is involved in the growth of neonatal cells. Yeah, I believe him. So now the question is how do they... here we go. So what happens when you knock it out? All right. Sounds fun. All right. So let's see here. CPT1 induces hyperplastic and hypertrophic growth. What about 1A? They're looking at 1B, but 1A was the one that had the counter, a reverse in the expression. Actually, now I'm a bit curious. What's the difference between 1A and 1B? All right, back to figure 1. So yeah, 1A and 1... there it is right there. I just missed it. Sorry about that. I have an eye exam to schedule, and they just called me out of nowhere. So anyway, let's continue with this paper. All right, Okay, so let's see here. So yeah, they looked at 1A, 1B. They show that there is an increase early on, and then a continued rise in CPT1B over time. I'm actually quite interested in 1A right here at the beginning. It rises and then goes back down. What is going... I wonder if they will talk more about that there. I am very curious about that one. I'm very, very curious about that one blip. Because they then end up focusing mostly on here's Eto mixer, which would be a stain. Or wait, what is Eto mixer? I think we're going to have to look this one up. There it is. Yeah, it's a treatment. I thought so. Told enhanced incorporation of EDU, increased number of cells, and elevated protein levels of cyclin E1, and strong expression of NAPB and ACTA1 markers that are highly expressed at embryonic and fetal stages, but are downregulated after birth. All right, so it's pretty much it's pretty much a treatment to help with this. All right. So yeah, here they are showing off some more of the data. These are very pretty pictures. Check these out. Ah, they're very pretty. Yeah, there's pH3, suractin, DAPI. DAPI is a nucleus stain. And here we can have EDU and again, the 3 stains again. So we have an increase in the when you have ectometer in all 3 cases. Here we have this should be their knockout. Yeah, showing that off. Upregulation, upregulation. Oh, it didn't load. So yeah, pretty much they're just showing off. And again, we quickly just mentioning again, they're in figure 1. They're pretty much just showing off the fact that they saw that the CPT1A and B, they are regulated differently. They focusing on B. They show that they have a knockout, which will increase, which has changes within the levels. For instance, here, CT1B, when you have their knockout, you can see that they have absolutely no expression, which means, boop, they knocked it out. By doing so, they're able to completely... Oh, it looks like A is elevated. Interesting. I guess A is elevated again. And then they just stop looking at A altogether, don't they? I am very curious about what is going on with A. CPT1A. Maybe it'll be in another paper. And then here you can see the hearts are enlarged. Oh my, they have gotten bigger. That is not healthy for the mouse. That can't be. Okay. It's definitely a bigger in cell surface area and mass. All right. Interesting. Okay, so what about the next figure? So this, I guess, is just showing off the different levels. So this is how they generated the knockout mouse. Here is the HW-BW ratio for the 10-week-old. The Cree, which is the control mice. Oh, they used the Cree line. Okay, so Cree mice. There's many different strains of mice. I guess I should probably preface that first. There are many, many different strains of mice, and the reason why is because they all... In order to make sure what we're seeing in science is accurate, we actually created a ton of inbred mouse lines. And I mean that they bred siblings upon siblings of mice to pretty much tons of inbreeding of these lines to make sure that all the mice we get are all genetically the same. And the reason why you do this, as I mentioned, is because if you don't, then there could be potentially a lot of variability in your data just because of genetic differences. So it's always interesting to see, for instance, which type of lines they use. They particularly... Let's look up the Cree mouse. Oh, the Jack's lab. Oh, the spot where everybody gets their mice. Let's see here. I think, here we go. Ah, they're using, when they say Cree, they're talking about the Cree recombinase. All right then. But there should be another mouse called Cree. Or my brain is rotting. The last time I've looked at a scientific paper was like years ago. So here, the Cree recombinase line. All right then. It's good that we looked it up to make sure that what we're thinking is accurate. So the Cree-LoxP mutant system. So this is how they ended up generating their mutant lines. All right then. So if that's the case, then yeah, Cree. So this line Cree is pretty much saying it's the mouse with the Cree recombinase inserted, but they haven't done anything. And then this is where they use the Cree recombinase to cut the gene out. Well, this is quite useful. If I recall, the Cree-LOX-P system allows you to cut it out after development has occurred. Tamoxifen. Yeah, inducible Cree-R-TAM system. Yeah, right here. So yeah, here we go. An explanation of the system right here. So pretty much we have, once you put in tamoxifen, you end up having heat shock protein 90. You end up resulting in the Cree-ER heat shock protein 90. This protein complex, tamoxifen causes the cleavage, which will result in the breaking of Cree and ER. That can then enter the nucleus and proceed to cut out wherever they can find the promoter gene. By doing so, you can effectively cut the genes out. This is very useful. Yeah, here we go. The Cree-LOX-P mutation system. This is actually very useful, especially in models, especially in these types of models, organisms, and systems. And the reason why is because of the fact that a lot of genes out there that are available, they are very important in development. A nice example is there's a gene called Wnt. Let me load that up, show it to you as well. Wnt gene. There we go. The Wnt signaling pathway. The Wnt gene is a very important gene. It is involved in development. It was originally, the reason why it's called Wnt, right here, the reason why it's called Wnt, it was because it was discovered in flies. The flies pretty much were wingless when you knock this gene out in these fruit flies. Oh yeah, right here, you can see it. Wingless flies. Yeah, so this is where the Wnt came from. However, they found out that when you knock it out in animals, yeah, it's a highly conserved gene in animals. And when you knock this out, I believe it causes, let's see here. Emerald fruit flies later is found responsible for amyloid flus and breast cancer. Oh, no. Come on, where is it in development? Because if I recall, it ends up resulting in, I believe if you knock out Wnt, it causes you to not develop a head. Yeah, I believe that if you target Wnt, yeah, repressor of activity of headless. Yeah, there's a gene called headless. Okay, all right. Because I remember I actually did study this pathway quite a bit, which is one reason why I remember it, the Wnt pathway, and that it's wingless. And this is why I'm trying to remember Frizzle. Some of the scientists, they just love their names of their genes. But yeah, I believe. Anyway, let's get back to the main topic. The main point is if you knock this gene out during development, you will produce nothing but organisms that cannot survive. They will be headless. They will have such a defect that they will only survive for a couple hours or a couple days outside of the womb. So because of that, it made it quite difficult to study certain genes. And the development of the Cree recombinase system, this Cree-LOX-P system, was designed so that you could get the organisms past development, past the point where they desperately need these genes, and then you can knock it out. Then you can knock it out and see what they do. So I have a strong suspicion that CPT1B, because they use the Cree recombinase system, might be involved in development and other. It might be a rather important gene. But either way, so here's their recombination. They end up creating the mice, so then they can then knock it out once they need to. And then here you can see the knockouts. So here's Cree, here's our knockouts. This is the weight index that we saw earlier, which is showing that the hearts are generally larger. We have an increase in EDU, which is a proliferation marker. And then KI67, this is the actin. So this is an increase in this complex. Likewise, we have an increase in pH3. This just increases across the board. Ooh. Increases across the board. And then likewise here we can see TAM increases. Let's see here. What is exactly is TAM injection? TAM treatments. What are ye TAM treatments? Let's go up to the methods paper section. Here we go. So in experiments, the genes activated by recombinase combination, corresponding Cree recombinase expression strains out of the flock's target genes were analyzed and used in negative controls unless indicated otherwise. CMC control mice were subjected to the same TAM treatment as the actual gene inactivation experiment. All mice were maintained in the... so we need to find another mention of TAM. Oh, they don't mention it. Oh, come on. Why do you not mention it? Alright. To Google, or DuckDuckGo, because I can't trust Google. Transient abnormal... Here we go. Oh, it's a cardio. Alright, it's cardio. Boop. It is a cardio term. So it's a transient abnormal myelophysis. This is in Down syndrome. All right. So what we want is a tumor associated. This is why I like to kind of know what my acronyms stand for. Here's transient abnormal myelofiliosis, but then here's a tumor associated macrophages. They're both TAM. All right. So which one is it? Treatment of TAM. Okay then. Okay, let's see here. Maybe the paper will... This is why I don't like acronyms. This is exactly why I do not like acronyms. TAM. Where can I find TAM, the particular... where this acronym stands for? I should probably put a space there. So it's a TAM injection. Okay, so we know it's an injection. And the reason why is because science... It's just basic mathematical calculations. You have three letters. That means you can have three letters when it comes to an acronym. Just out of the alphabet, it has to be 26 times 26 times 26. So I'm getting out the calculator to figure this out. How many random combinations do we have? Let's get out our calculator. So 26 times 26 times 26. So that's 17,576. So 17,576 combinations of the the English Roman alphabet. There are over 1 million words in the English lexicon. There's just no way you can have this combination of words without having some form of overlap. It's just not possible. So because of that, it's very annoying when you start getting these acronyms everywhere, because then you're like, okay, well, what does this acronym mean? It's going to mean something different no matter where you go, and it's doubly in science. The fact that they say TAM... let's see here. So we have injection. So let's type that in. TAM injection. Let's find out what this is. Optimize tamoxifen induce. So is that what it is? Tamoxifen is the TAM tamoxifen injection. It is. Ah, there we go. Tamoxifen. All right then. I probably should have guessed, given that we're using the Cree recombinase system, but you always want to make sure. So TAM tamoxifen. Actually, you know what? Where's tamoxifen in here? I want to do a quick check. Tamoxifen. There it is. Aha, okay. So yeah, they do mention it in the methods. All right. So they do at least highlight what it is. Good to know. Anyway. So yeah, I just need to read need to keep an eye out. Anyway, let's get back to figure 2, which we're looking at. It's right after figure. It's right after figure. All right, so it's right. it's quite low. No wonder why I didn't see it initially. Usually when in these papers, they like, because, okay, this is not a, this is just a nitpick on my end because of the fact that it's always easy for readers. But usually, whenever the first time it shows up, they usually like to do it, whether it's in the figure or whether it's in the text, because most of the times people jump around within the papers, like what I'm doing right now. And the reason why is because papers are extremely dense. They're ludicrously dense. So just kind of going through, looking at these and figuring out what's here and what's there. This is, you tend to jump around and you tend to see what you want to find, what you're interested in, and you'll miss things. So usually when you end up going, all right, here, and then you see TAM injection, particularly right here, you'll see TAM right here, figure J, TAM. Usually they would put it somewhere in here because this is kind of the first time you would see TAM, but they chose to do it within the paper. They chose to do it within the wall of text. So, okay. Either way. So, tamoxifen injection, they ended up seeing that they managed to have a increase in the heart rate condition, which is what's typically seen when you knock it out. And we don't get it in the control, which is basically just showing us that tamoxifen is not causing the increase in the size of the heart. And then likewise, the same thing going on. Tamoxifen, here we have the various other treatments, just kind of showing off that tamoxifen is not causing any problems, which is very important. You need to have your controls. And they showed off all their controls, which is nice. And then last but not least, they show cell numbers between tamoxifen 4-week, tamoxifen 8-week, and there is a significant increase in the numbers in O. So, oh, hey, Tukey. Aha, Tukey tests. This is a statistical test. It's a Tukey-Kramer. It's pretty much a student's t-test, but it is a it's pretty much a ranked student t-test. So they did end up seeing a significant difference here, which I wonder if they'll comment about that a bit later. Anyway, next figure. So deletion of this reverses the cell cycle arrest. So this is where they end up seeing that here. All right, let's load this figure. Let's load this figure up and get a nice closer look at it. Let's see here. Let's get a nice closer look at this figure. Let's get all in here. Let's get all in. There we go. So here's our nice 3 stains that we've pretty much been seeing for quite a while now. And wow. Okay. So here's the control, and then here is the knockout, and then they have a stain right there. Just one though. That's a wow. That's just, what about this one? They found another one right there. I wish that would load. Or come on, load. I want to look at that. There we go. Oh, they found 2 here. And then here, they also found some more staining in the knockouts. All right then. So they're seeing it. They are seeing an increase. It's not that big of an increase though. Let's see here. They take their mice, they inject EDU, they then isolate the nuclei, and then they perform the staining. And then they use a fact sorter. Probably wise, given that, well, the heart is a bit larger. And it looks like there are very few cells, so it makes sense to do the fact sorting. And then here we have the trichrome staining. Control, knockout, all right. Interesting. So structurally, it looks fine. And then here we can start seeing that there's actually not much differences in the levels, huh? Yeah, there's not much different in the levels. Oh. That's a bit interesting. That's a bit intriguing. Let's see here. EDV, ESV. What do those two mean? EDV, ESV, where are you? Let's see here. This happens to be, we're looking at O. M and O, here it is. Oh, MRI analysis of the left ventricle ejection fraction, which is the LVEF, and the endocytosolic volume. EDF is the, see, this is what I'm talking about right here. This is what I'm talking about. This is what I'm talking about right here. They explain the acronyms right here. All right then, so endodiastolic volume. So they're looking at the different volumes of in the heart. And there isn't really much, wow, there's not much of a difference. There's not much of a difference in the volumes. That's, so the heart is just bigger. It's not pumping more blood. It's just the tissue, all of the cells are just bigger. Ooh, okay. All right, let's get back over here to our figure. So yeah, pretty much there's just more confirmation that the heart sizes have grown, but there is no significant differences in the spaces of the heart. So where the fluid is, where the blood pumps, there's no differences. TAM4, TAM8. So this is 4 weeks after after cutting out the gene, 8 weeks after cutting out the gene. So there is a significant increase when they cut it out, or when they cut the gene out. So it's definitely involved in the in cell size, but it's not really having a major impact on the heart flow and heart rate. But when you do knock it out, you start okay. I've been seeing Ki-67. Yeah, I've been seeing Ki-67, thinking, oh, I know what that is, but we haven't actually defined it yet. Ki-67. All right, so what do you do? We want to find the methods. All right, to the methods section. If we can find the methods section. Activated Ki stimulates the confirmation that accumulates of alpha Kg is a critical signal to prevent CM maturation and enable proliferation. We treated postnatal at 4-day with a cell permeable alpha Kg. We observed a marked increase in Ki-67. So it's a marker for is it a proliferation marker like we were thinking? All right, when it happens like this, just look it up. Okay, why is the all right, why is this anyway? Both a bit different. Don't know why. There we go. Ki-67. But this must be it. Antigen is a nuclear protein associated with cell. Yep, it is. I was right. I knew I wasn't crazy. It is a cell proliferation marker. Okay, so yeah, so this is just a different cell proliferation marker that they're using. So here they're showing a proliferation, and then pH3, which is also a cell proliferation marker. And then I'm guessing, that if I looked it up, Aura B is also a proliferation marker. So they are getting increases in proliferation. How much do you bet they're going to show it off in the next figure? Let's go. And are these the counts? Abrasions reverts maturation of CMs, enriched biomedical processes. Wait, where's the where's the calculations for the increases? Oh, it must be these, right? Well, this says relative expression. This is body weight. Okay, they showed us pictures. Where's the numbers? Extended data. How much do you bet is in the extended? There's I don't see it. How much do you bet? It should be kind of showing up though. Oh wait. Did my brain have a brain fart and forget? Yeah, they already showed the increases earlier. Duh. Yeah. Okay. Yeah, they already showed the significant increases. This is just kind of confirmation. Yeah. All right. Yeah, it's good to always go back and check. Okay, so yeah, so what do we know? So what do we know so far from the first two figures? Well, we know that this gene is important in development because they're using the Cree recombinase system. We know that when you knock out CPT1B, you have hey XO, how's it going? We're going through a paper right now. Yeah, it's been a while. We're having fun going through a paper. Yeah, so let's see here. So we know that CPT1B is important in cell cycle. It's a fatty acid oxidation cycle. It also is upregulated when you have an abrasions or damage to the heart. So they created a model to knock it out. We also know that knocking it out results in increases in proliferation. We see it through cell counts via stains. And we also know that it increases the heart. So the size of the heart increases, but the amount of flow, the interstitial or these areas in the heart where the blood flows are not increased in volume. So we know that. So we have this information in hand. So let's see here. So what happens in the next figure? Yeah, right now we're having fun going through a paper, trying to figure out. It's going to be fun to go through a paper. So we're going through a paper. I wanted to go through a paper, so we're going through a paper. So here we go. So more gene expression right here. The z-score of 1. Whoa, okay, the number is going down. Okay, the number is going down. All right. Again, usually you want this to be 2. Plus and minus 2. The fact that they're going to 1 now tells me that the gene expression is quite... they're definitely going to have to do some extra work to make sure to prove that these changes are important. Because once you get below 1, as I mentioned earlier, once you get below 1, then suddenly these might just be normal fluctuations that happen. So it's you have to be very cautious with these numbers. You can get away with it when you have time series data, but when you're doing this single day time point data like this, it's sometimes you have to take it with a bit of grain of salt. Let's see here. So we have a knockouts. So control, knockout, control, knockout. And then what's the difference between the left side and the right side? C knockout and I knockout. Ah, okay. What's the difference between the C and the I knockouts? They created 2 different recombinase knockouts. Well, I mean, I thought they only had the 1. All right, let's go. Let's see here. This is a term enrichment analysis. So, okay, so all right, they can get away with an expression of 1 then. Because this is a Go enrichment analysis. This is different from gene data. So enriched biological processes for upregulated genes are in red and downregulated for genes in blue. Heat map of selected gene plus process upregulated in the red and blue. So maturation, concentration, HIFA signaling based on z-score normalization of the sequence. Oh, no, it is sequence counts. It is sequence counts. So in other words, they are in this is a bit of a tight squeeze right there. I wouldn't complain though. Again, data is messy and dirty. So it's just me. I as a biologist, I would take some of this with a grain of salt, but this just means that there's more targets to potentially look into. Because as I said, sometimes gene changes are so small that you can't really see them with the extreme changes. And sometimes it's these minor gene changes which are the most important. So let's see here. And it looks like they ended up doing some fold change analysis right here. Yeah, oh, here's some downregulation, which is quite interesting. Here's some severe upregulation. This is all on a log scale too, so wow, quite crazy, quite crazy. All right, and then here's some overlap for the amount of genes that are expressing. And then the biological processes as well. Heart developments, positive regulation of cell differentiation, muscle cell differentiation. There's a lot of different Yeah, this looks awful lot like what my paper used to look like. Oh, this looks an awful lot like what my paper used to look like. It's because when I published, one of the reasons why I was gravitated towards this paper was the fact that they were doing a lot of high-seq analyses and looking at gene expression changes, and that's kind of what my forte was when I was a scientist. So yeah, I looked at a lot of these things. I tried to generate a lot of this data. You won't believe how many sequencing experiments. Oh, it was so many. And because of the fact that you had to schedule it and the fact that I was on a timeline, I would start doing my sequencing at like 6 o'clock, 6 p.m. All of the sequencing, because it was all RT-PCR. So it was real-time PCR that I was doing. And you'd start it at 6, it would end at 9. You had to collect the data and then clean up as well, because guaranteed at 6 in the morning, someone would be using the machine. So you had to get your stuff done. So started at 6 p.m., finish it off at 9 p.m., clean it up. So I'll be going home at not even going home at 10, going back to the lab at 10 so that I can then document everything and then leaving at 11. So yeah, I even remember in those 3 hours I was waiting for the RT to finish off, I would be generating these things. So I remember generating these quite a bit. I believe there's a tool available. Oh, sadly I don't remember it, but I do remember that there was a tool available to those who were using the sequencing lab, the sequencing department, or they call it the sequencing lab, but it's really a department which primarily focuses on sequencing either through RNA sequencing, gene chip sequencing, even RT-PCR sequencing, all of these. So they had a free software, and I use this frequently. So I remember generating these. I forgot the name of the software, sadly. But nonetheless, the fact that they have a lot of these basic ones, heart developments, muscle cell differentiation, this makes a lot of sense to see a whole lot of weird things, like in utero embryonic development. But then right here, the one that they're interested in, positive regulation of cell differentiation. Oh, that's very nice to see. That's why they highlighted it. All right, so that pretty much shows. So we have CPT1B. Knock it out, you end up having an increase in cell differentiation. And this is quite important, quite important. Next figure. Actually, this is all in the heart. I'm actually very curious. I'm wondering, what does it look like in other regions? If I was still in research and I was studying cell development, especially in a mammal model, I would definitely be looking and going, CPT1B. I wonder what it's doing in other organs. Could I get cell differentiation maybe in the nervous system? Could I potentially, because, there are many parts of the body, the human body, which, they fail significantly. And I mean, they fail significantly at regeneration. Not every tissue can be the liver or the skin. So I'm wondering if CPT1B would have an effect anywhere else. So let's check out here. What's about the next figure? HIF1A, all right then. We have an upregulation following knockouts. Tunnel, ooh, tunnel. Tunnel's also a stain. I believe tunnel is also a stain for I believe I use tunnel quite a bit for proliferation. I use it for a proliferation marker. It gets incorporated into, so the way tunnel works is it incorporates into your the DNA. So it's a DNA replication marker. So what happens is DNA will start being replicated. You have tunnel, which will then stain, which will be incorporated, and thus, as the more you have cell differentiation occurring, more and more tunnel cells will show up. Okay, let me make sure that I'm not remembering this wrong. Yeah, exactly. It's labeling for oh, it's a DNA defragmentation. So what was I thinking of? Oh, what was I thinking of then? This is for DNA defragmentation. So that means this is for identifying cells that are dying. Okay, so what was the other one? So yeah, tunnel is cell death. So what was the one I was remembering? Because I used both. It's been almost 10 years since I used it, which is... So yeah, this is... I remember to involve DNA, so it's not... Let's see here. Proliferation marker... Let's see here. What are the different common markers for proliferation? There we go. Oh, come on, nature. Don't be like that. Let's see here. Tunnel is cell death. And then we had... PCNA. I don't think it was PCNA, was it? I don't think it was PCNA. Okay, let's do a quick peek at PCNA. I'm checking this out to make sure that to refresh my brain. Is DNA clamp that acts as a proactive function of DNA polymerase. PCNA is a homotrimer which activates its primarily circling the DNA, where it acts as a scaffolding to recruit proteins involved in DNA replication, DNA repair, chromatin remodel, and Actually, maybe it is PCNA that I was thinking of. Because as you end up getting more DNA replication, you'd need more scaffolding, which means you'd have an increase in PCNA. It also can be used as an indicator for, as I mentioned, chromatin remodeling and epigenetics. So, okay. PCNA, all right. I'm glad we checked. So, tunnel, cell death. So here they're looking for cell death. So this is the DNase, this is the control, and this is the knockout. So DNase. Maybe I should have picked those. Maybe I should have picked another model that I'm more familiar with. It doesn't matter. We're having fun reading paper. And the more we go through, the more we'll remember. So B. From 25-week-old control mice and knockout mice, DNase treated samples were used as a positive control. So DNase, I am guessing, is something that just breaks up the DNA. It's a positive control, which means we're supposed to see tunnel. And they have a whole lot of it. And in the controls, wait. I think in the control there's a little bit, if you look here, there's a little bit of red. Don't you see it? Here, and here, there's a little bit of red. And in the CPT1B, there isn't as much. Likewise, hydrogen peroxide treatment. Oh, that looks nasty. Okay. Hydrogen peroxide treatments, followed by the controls and the MFI. So we're looking again at the MFI. There's no difference. And then here we have another stain. H2AX control at CPD. We have a decrease. All right. I'm surprised it didn't give us the the tunnel calculations. It might be in a supplementary data. They do have extended figure 4, which, oh wait, is this we are in extended figure 4. Okay. So that means this is supposed to be an extension of figure 3, oh boy. I think we might going to have to read through the results to kind of look through it to kind of get an understanding of the flow of the figures. Usually it's easy for you can just look through the figures and instantly pull it out, but I'm asking for certain data and I'm like, oh, where's that data? Thinking, and then most likely it's in a different, because extended figure 4 should probably be a supplementary for a different figure. Yeah, here we go. This is what I'm talking about. Figure 3 and extended figure 6. So that means extended figure 4 that we're looking at has to be a supplementary to another figure. Which one, though? 2A, so that means it's probably, here we go, extended 3. Extended 3, extended 3. Extended 2. Oh, so it's kind of its own figure. All right. Next, we carried out RNA-seq analysis and isolated the controls. Different term enriched analysis recognized by upregulated genes associated with fatty acids and lipid metabolic processes in both mutant mouse lines, most likely reflecting an attempt to compensate for abrogation of FAO. In both mutant mouse lines, we also detected strong upregulation of genes with cell cycle progression and reduced expression levels of genes involved in maturation, concentration, concomitance with upregulation of differentiation markers. Downregulated genes were primarily, oh yeah, this is the figure 3. So we want to check, yeah, extended figure 3. So figure 4, right here. We did not detect differences in the levels of reactive oxygen species between control and the knockouts, indicating inhibition. The inhibition of FAO does not attenuate generation of reactive oxygen species. Nice. However, the percentage of YH2AX cardionuclei dropped the cardiac nuclei dropped markedly in the deficient mice compared to control hearts, suggesting that inhibition of FAO reduces DNA damage, irreceptive of reactive oxygen species, or stimulates DNA repair. Taken together, the data indicates that inhibition of FAO initiates a cascade of events converting adult cardiomyocytes into the more immature state that enables cardiomyocyte proliferation and to a lesser extent favors hypertrophy. All right then. That explains a lot. Probably people that go, "Oh, what does that explain? What does that explain? Explain." All right, so pretty much what they're referring to is that here we have a marker for the nuclei, and we actually see a significant decrease in that. This so pretty much this is kind of what we're seeing here, right here. We have a significant decrease between the control and then the mutant. However, this is completely different when you compare, or when you look at other markers, you tend to see that there isn't as much of a difference, which is most likely referring to the fact that we're, even though we're causing damage, we're not resulting in reactive oxygen species, which are they're responsible for DNA damage, for causing lots of problems. Usually in high stress environments, you have an increase in reactive oxygen species, these ROS particles, and as such, you can result in lots of damage. This is one reason why one reason why you tend to see people say, most medicines nowadays say that you can take this medicine, you can it will help reduce reactive oxygen species. I think there's a lot of supplements right now which claim that they do this. And the whole idea is because if you have an increase in this, you have an increase in damage, which decreases your chance of recovery. So by decreasing this, it will result in more recovery or easier recovery. And the fact that they end up they don't see a increase in this suggests to them that some other avenue is being incurring when it comes to the proliferation and to this proliferation of the cardiomyocytes. And they also show that inhibition of FAO. Let's see here. Let me make sure we have FAO figured out. Oh, okay. I don't get why copying FAO is so difficult. Let's see here. Role of FAO. Here we go. Fatty acid oxidation. Should have guessed. So this is the fatty acid oxidation. Oh, that actually helps out. That actually helps out understanding this figure a bit more. So right here, let's go back to this paper. This percentage of the cardionuclei dropped markedly in the deficient mice compared to controls, suggesting that inhibition of fatty acid oxidation reduces DNA damage, irrespective of ROS, and stimulates DNA repair. So this is kind of the hallmark of the paper. Yeah, I tend to have trouble holding on. This is what, again, I don't like acronyms. I keep forgetting. But yeah, so pretty much this is showing that because the fatty acid oxidation is what's causing damage and preventing the cell cycle progression. This was what they showed off in the first few figures. And here now they're showing that when you have your mutant, you have a decrease in the amount of the fatty acid oxidation. So you have a decrease, which is good. You have an increase in proliferation and repair, which is good. However, you do not have an increase in reactive oxygen species, which could have potentially been a second avenue for damage. This could be what's causing oxidation, these reactive oxygen species. But because you don't have that happening, you don't have these ROSs causing oxidation, and you have a decrease in the amount of fatty acid oxidation, it tells us that there's a pathway that's being inhibited by, CPT1 is involved in a particular pathway, which is what's causing these oxidation of these fatty acids that is not through ROS. So that means that most likely, as I said, there's another pathway involved. And then this is kind of highlighting this. Let's see, what is this? This is figure 2. So this is kind of the more significant data of what was being showed off in the extended figures. I kind of wish the extended figures were at the bottom. This is a complaint about how they organized the paper here. I kind of wish it was at the bottom because it would make it easier for my brain to here are the main figures, and then they mention link to the supplementary figures. But it's just how the people like to do it. But anyway, here we go. Increase in the heart rate, increase in heart, increase in heart. Here is so these are the different times that they did this is the different methodologies they used. So here we go, trichrome staining, quantification, a fibrosis scar area. Oh, A into D is A, B, C, D. This is ooh. All right, A, B, C, D. A to D, this is the trichrome staining. A and C are the quantification of the fibrosis, which is the scarring area, and B and D are the heart sections from the control Cree mice and the knockout mouse. The control mice had 9 and the knockout had 7, which is pretty good numbers for mice. So control. So let's see here. So this is the stainings, the trichrome staining. And this is the quantification of the fibrotic tissue. So in our knockouts, we have a decrease in the fibrotic tissue. So here we can see that here and here. Let's see here. What's the difference between... this is both B. So this is total fibrotic area, and this is total fibrotic area normalized to heart size. Got it. Because the heart was bigger. So they had to make sure that we understand. It's not because we don't see a decrease. Well, actually, technically, yeah, we don't see a decrease because of the fact that the heart is just generally bigger. You know, it could be equal sizes, but because of the, but you kind of see that with the total fibrotic area. But again, the arguments would say, it's fun. I understand why they did this, but I feel like having just one of them would be enough. If there was an increase, then yeah, definitely need that control. This is just kind of hammering it away, like, you know, to prove to a point that this is purely because of the because we knocked out CPT1B, not because of any other abnormalities. So here we go, and then here's another one, more fibrotic area, and then here we can see this is our treated group with IR, and then this is with the TAM. So this is control TAM. All right. And this is more fibrotic area with their knockouts. So I think this is just different knockout models. Nice. Oh, very nice. That means they have different methods for knocking out CPT1B, which helps explain, because if you remember, I was commenting earlier about they had two different models, but I was unfamiliar with the other one. So here it helps explain it more. That's what happens when you put the extended figures first. Anyway, but good. So it's good to see. So you have two models. One is the Cre recombinase. Another one is another one is this I knockout. I wonder what the I one is. The main point is they have two different models, and then when you knock them out, we see a decrease in both. And then likewise, the AAR. There is no significant difference between the two, but the IF of the AAR, there is a significant decrease there. So let's see here, EF and then G. So here we go, EF AAR is the left, and then the IF is the infract area of the AAR. So this is one of the regions in the heart. So in other words, yeah, we have a significant decrease in the amount of, see, AAR, no difference, but the IF of the AAR, a significant decrease. So the infracted area, which is the scar tissue area, the area that's been damaged. So yeah, this figure right here is pretty much showing they are in fact regenerating. That is actually something that is occurring. All right, nice. And then this is their methodology. And then more data. So looking at more fibrotic area, again, we see a nice decrease. And then here is a ratio of the LVEF, the LVEF. Left ventricle injection fraction. All right, so the left ventricle, so this is looking at the left ventricle. And we see a marked improvement. What do you mean that there's no, oh. There is no significant difference in the control, significant difference in the knockout. So yeah, we're actually getting heart regeneration. Wow. We're getting heart regeneration in mice. This is very nice. Oh, that is very, very nice. And the numbers, let's see here, N of 6, 5, 4, 5. The numbers are also quite nice. Ooh, yeah, the numbers are quite nice as well. So the fact that they're getting such significant calculation. Oh, actually, we should do a quick check. They're doing a two-way ANOVA with a Tukey-Kramer correlated. All right. So yeah, they're doing it. I think that's quite nice. It's a two-way ANOVA, which is quite good. So for two-way ANOVA is pretty much think of an ANOVA as a multi t-test. So pretty much when you have more than one condition, you have to kind of figure out which. When you have more than one condition, you want to try and figure out which is significantly different from the group, and that's what an ANOVA is. It's pretty much a t-test that looks across multiple conditions, not just comparing two different conditions. And as I mentioned earlier, Tukey-Kramer is it's pretty much a ranked t-test. Wait, sorry, Kruskal-Wallis is a ranked t-test. What's the Tukey-Kramer again? Oh wait, Tukey. Come on, Tukey-Kramer. There we go. The Tukey's range test. Here's the Tukey-Kramer test. I am right. The Kruskal-Wallis is the ranked ANOVA. Oh yeah, okay, my brain still isn't turned into cheese yet. Okay, so yeah, the Tukey-Kramer is a